For today's lecture was alteration of immunological parameters during HIV infection. It's a very straightforward topic, and the reason why I chose this topic, two reasons, because I have been, as mentioned by Dr. Gopal, that I've been working in HIV since last more than 15, 20 years, I would say. Initially, I was working as a postdoc, then I came here in GNU, started working on the similar topic, looking at different aspects. Another important feature is that key immunological parameter, why they are important, because the reason why I chose this particular topic, that to discuss about immunological parameters during HIV infection, so that you should know the importance of immunological parameters, because once we know about different kind of immunological response during HIV infection in the initial phase or later in the chronic phase, then you'd be able to know that which immunological parameter is beneficial for stopping the HIV disease progression, or which is which actually is harmful for the infection, so that you could then come up with the new therapy, or come up with a new therapeutic approach, so that you can modulate the system. Anyway, so you see that HIV infection, we just talk about Indian context. Globally, you would be surprised to know that there are still 39.9 million people living with HIV, and India being a third largest HIV epidemic so far. And if you see the data from previous years, you will find that people living with HIV in India, 2.5 millions. Globally, there are 39.9 million, but in India, there are 2.5 million, approximately 25 lakhs people still living with HIV per year. I mean, this is the data from 2023. So, new HIV infection took place in India around 66,400, and 22 data AIDS related deaths were reported in India, were 39,600. So these are just statistics of the HIV and in India. But when we talk about HIV and how it actually infects the cell, so HIV needs a receptor. The receptor which we talk here is the CD4 receptor. I just select the laser pointer so that you get to know CD4 receptors. So it infects all our CD4 T cells. But in addition to CD4 receptor, it also requires CCR5 or CXCR4 as a co-receptor. CCR5 receptor, when the virus uses, is called R5 strain. If the virus is using CXCR4 as a co-receptor, it is called X4 strain, right? This is the life cycle of HIV. So when HIV infects CD4 positive T cells, it releases its nucleic acid, which is RNA. RNA is reverse transcribed and forms double-stranded DNA, and this DNA gets inserted in the host DNA itself. So it makes a part, the host DNA mixes or recombines with the viral DNA, and then this viral DNA gets integrated in the host nucleic acid or host DNA itself, and this what we call provirus. And the provirus, this performs the process of transcription. As a result of transcription, new RNA comes out of this viral DNA. And from this RNA, the translation takes place from RNA to proteins. Polyproteins are formed. And this small viral proteins, they get assembled. And the new viral particle is released from this CD4 T cells. Now, this cycle continues, and then we end up getting so many viruses, and they keep infecting other CD4 T cells, and that is how disease progression or HIV pathogenesis takes place. This is a very important slide. I'm not discussing about how HIV spreads from one person to another person. That is a different topic, since we have to be restricted towards immunological response. So it's better to look at the HIV pathogenesis, what happens. You see on the x-axis, we have times like weeks followed by years. On the left y-axis, we have a CD4 T cell count, which is in the blue color. On the right side y-axis, we have HIV RNA copies per ml of plasma. So suppose when a person gets infected, virus enters in the body, what will happen? Let's focus on this red color line, which is a HIV virus or HIV RNA. It starts increasing. It increases until it reaches at to the maxima. That is the highest HIV viral RNA load during early HIV infection. When it reaches at the peak, then slowly it starts coming down. Why it is happening? Because virus comes up, starts proliferating, and then your immune system gets activated. When your immune system is activated, at that particular stage, the virus has not mutated at all. So the immune system is able to bring the viral load down. So viral load starts coming down, coming down. But while it is coming down, it is not completely eradicated. Some threshold remains there, and this threshold remains in our body for several months to years, and this is what we call clinical latency. On the other hand, with this blue line you can see, which is a CD4 T lymphocyte on the y-axis on the left side, as viral load is increasing, viral RNA is increasing, your CD4 count starts decreasing because it is infecting CD4 cell, and CD4 cells are getting depleted because of so many different kind of effects, which is pyroptosis, cytopathic effect, and so many other mechanisms. And then CD4 starts coming down, 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 down. But you can see that the moment viral load starts coming down, then there is a restoration of CD4 count as well. And during this phase, what we call clinical latency, these two things goes parallel. But later on, after several years, what happens? You get some sort of mutation in the virus, as the result of which your immune system is now able to not able to control the viral replication, as a result of which virus shoots up. You can see that, and your CD4 count comes down, down, starts coming down, and then you reach to a phase where you can call a person with opportunistic infection that this person has got AIDS, acquired immunodeficiency syndrome. So now, based on that, there are three different categories of HIV infected individuals. One I have listed here as a non-controllers. Non-controllers are those people who are not able to control the virus. It means that in this latency in the clinical latency phase, which I mentioned that it goes from several months till several years, the median viral load is approximately 30,000 copies per ml. Now there are different category of people, HIV infected subjects, who are on antiretroviral therapy. When they are put on antiretroviral therapy, the viral load goes below 50 copies per ml, or sometimes we call it undetectable. So most of the people who take antiretroviral therapy, they are called ART suppressed, where the viral load is below 50 copies per ml. Third category, which is only 1% of all HIV infected subjects, we put in this particular category, it's called elite controllers. Elite controllers are those people who do not have detectable virus in their plasma, and they have preserved CD4 count. CD4 count is also not depleted. And this is happening in the absence of therapy. So they do not require any therapy. They are born with some kind of what you can say genetic feature, which makes these people elite controller, that they are surviving. They are completely normal, like a person with the normal individual who do not require any medication, because they do not have detectable viral load, they do not have any issues related with the HIV per se, but they do seem to have other kind of complication I'll discuss later in coming slide. But just remember elite controller, because I will talk about the immunological responses which are developed in elite controller as well, because you see the idea, elite controllers, they are fine. So the kind of immune response we can observe in these people could be very beneficial for us, because we know that this set of cytokines, this set of T cell responses, they are very good. And if we are able to mimic that response in non-controllers in the very first category, then what will happen? You will be able to control the viremia. We will be able to control the disease progression, and we can have a better quality of life for those HIV infected individual who are non-controller, right? When we call a person with AIDS, with the acquired immunodeficiency syndrome, there are three things that needs to be there. A person has to be HIV positive, and their CD4 count has to be below 200 cells per microliters. And in addition to these two features, the person should have HIV related conditions or symptoms, like those morbidities, those sickness which defines HIV. These are nothing but opportunistic infection, right? So these are three categories. Now look at this picture again, the same kind of image which I showed a couple of slides back. Here on the y-axis we see weeks, years. On the x-axis we see weeks and years. On the y-axis we have several different parameters. Let's look at the one parameter which is the viral load. You see this HIV viral load where my cursor is there. Exactly following the same path, it goes up, 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 and then because of the emergence of the immune system, it goes down, and then it remains stagnant for a couple of years, and then it shoots up, and people develop AIDS. And how it is related with the CD4 count, you see that this red one is CD4 count, it comes down, 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 and then it becomes more or less similar, and then CD4 count goes down. What else is happening? You also have, you see that this purple color CD8 T cells, CD8 T cells emerges, and CD8 cells are also doing pretty well during clinical latency phase or asymptomatic phase. And when AIDS comes, it also tends to go down. And neutralizing antibody is also there, is very much present in asymptomatic phase, because person is asymptomatic because there are antibodies, there are CD8 T cells, but later stages they fail to contain the HIV, as a result of which HIV shoots up, CD4 count comes down, and the person develops AIDS. Here you see the three phases. The number one, 4 to 6 weeks, it is called acute phase. Acute phase happens right after the primary infection when HIV enters in the body. Acute phase is followed by chronic HIV phase. Chronic HIV phase, most of the HIV infection they are caught during chronic phase, because whatever symptoms or complication you develops during acute HIV infection right after the primary infection, they are not specific. A person with the flu-like symptoms may have had HIV from outside, but you will never know, because ultimately what happens, it's going to be okay, because virus will go up and then it starts coming down. Likewise, CD4 comes down and then becomes normalized. So you by default get into the chronic HIV phase, and most of the infections are caught during asymptomatic or chronic HIV phases. Now we have to find out what are the immunological responses existing. Two different phases. One is acute phases, very early phase, and what is happening during the chronic phase. It is also very interesting to note that whatever early responses you develop in the primary infection, they may also decide what will be your fate during the chronic infection, whether you will tend to go to the disease progression or you will somehow control the virus. That depends, or that decides your survival time, without any treatment, how long you're going to survive with this, in case if you're not taking any treatment. But everyone is on treatment these days. So how actually these things happens was, we need to know, when the virus crosses the mucosal membrane, if it is spreading through sexual contact, it will cross the mucous membrane, and what will happen, there are some dendritic cells. Dendritic cell captures the virus through their receptors called DC sign. And there are also free viruses. These viruses together with dendritic cells goes to the nearest lymph node. And the lymph node, you will have lot of T cells. And in T cells, there are also CD4 T cells. CD4 T cells, they get infected. True infection takes place in the lymph node. At day 3, you can imagine if virus is entering at day 1, And day 2 is the travel of dendritic cells towards the lymph node. And day 3 is the dissemination. It means that now these CD4 T cells which are infected, they will spread to different organs. It's called widespread disseminations: brain, spleen, gut associated lymphoid tissue. That's how HIV establishes its infection. And then people who get infection, they starts getting the symptoms, which is called primary HIV symptoms or acute HIV symptoms. One important thing here to note that when HIV infects a person or HIV goes inside someone's body, initial 10 days, you won't find anything. There is no way you could tell that the person got infected or not, because these small 10 days they call eclipse phase. This is eclipse phase. This is called a period of 10 days known as eclipse phase before viral RNA becomes detectable in the plasma. Right after 10 days, the very first thing that you will detect is HIV RNA. Now, once you detect HIV RNA, it is followed by detection of proteins. Until you detect the proteins, you already crossed 3 weeks. It means that 21, 25 days have passed. At this particular time point, you will develop early infection, acute HIV responses. What is happening during this particular phase? Detection of HIV RNA and P24 antigen in the blood before antibodies are formed. Acute human immunodeficiency virus HIV infection, AHIV call, can be defined as a time from HIV acquisition until seroconversion. What is seroconversion? Seroconversion is the term we use for the situation once your serum develops antibody against HIV. So first RNA came, and then protein came, and because P24 antigen, viral antigen, came in the picture, then what will happen? You develop antibody against HIV, because without protein detectable antigen, development or emergence of antibody is not possible. So once you have antibody, at that particular phase, you call now the person is seroconverted, because person has developed antibody against HIV. So this phase, virus enters, antibody is produced, is called early or acute HIV infection or early infection, right? So now, just to think about what is happening during this early phase, there must be some sort of cytokine or immunological responses. To study that precise immunological responses during this early phase, just talking about like initial 20 21 days, there are several papers, but what we am going to talk about the work which I did by myself. What we did, we were in touch with a blood bank which was a plasma donor, and many people were recruited, and we created a cohort we call Chavi cohort. And what had happened, we selected 10 plasma donor. We happened to have these plasma donor which used to come and donate their blood, and they were not HIV infected, but during sequential blood collection they got infected because of the lifestyle that they had. So remember, when they got the virus, immediately they won't actually develop viremia. When I say viremia, it means that right after getting infected, after how many days HIV is detected in your blood? It is basically 10 days, which I just mentioned, it eclipse period, but 10 days is a median. Sometime it is like a 15 days, sometime 14 days, sometimes 7 days. But for these 10 patients, we normalized everything. We had been collecting the blood sample, and these people became HIV positive. And we had a collection of blood sample pre-viremia, means before the virus was detected, and also post-viremia, when the virus was detected after this. So 2 time point before viremia and 5 time points after viremia. So once I had samples from these people, and then what we did, we did cytokine analysis just to find out the early immune responses, at least cytokine responses, very precisely. So you can see that on this particular figure where my cursor is. So I just mentioned this as a T0 point, T0 at the time when viremia was detected, the HIV virus was detected in the plasma. So two time points before and time point afterwards. 5 time points indicate 14 days to 45 days, right? And then we took a help from the previous findings, which was also published by our collaborator. You could see that IP10, IL-15, TNF alpha, all these top 5, 6, 1, 2, 3, 4, 5, 6, 7 cytokines are listed here. And it is saying that median day of initial elevation means that they were the early cytokines which were found to be elevated at day 6 post viremia. It means that when the virus came and after 6 days, right after the viremia, what happened? You saw the elevation of these cytokines. That was the published one. We wanted to explore some other biomarker which comes much earlier, and we ended up finding a very important molecule which is called galectin 9, which is a lectin which binds to the carbohydrate moiety of different protein, and it's a ligand for TIM3 exhaustion molecule, which I will discuss in coming slide. And we observed the initial elevation point, and we found that median day of initial elevation of galactin-9 was 5 days. It means much earlier than those cytokines which were discovered, which were found to be elevated at day 6. So that was our finding. And since galactin-9 is coming quite early in initial infection, then we wanted to see what actually happens in different categories of HIV infected subjects. So you see that here on the x axis, we have non controller, we have ARV suppressed, and HIV negative, three different category of individual. Two of them here, they are HIV infected for sure. One is non controller, it means not controlling the viruses. On the y axis, we have plasma galactin 9. So you can see that the median plasma galactin-9 level in non-controller is much, much higher compared to that of HIV negative control. But if the people are put on antiretroviral therapy, the plasma galactin-9 level reduces, significantly reduces compared to non-controller, but it doesn't normalize to the level of HIV negative subject. It means that despite antiretroviral therapy, the level of galactin-9 remains elevated. That is alarming, because what we were just thinking that if you don't have any virus circulating in the plasma, you should be perfectly all right, but it is not the case. Galactin 9 level is elevated. Likewise, several other parameters will also be disturbed. That is why people who are on antiretroviral therapy these days, they seem to be getting some complication every now and then, for which symptomatically they are treated, right? Now, once you compare your galactin 9 level with the HIV viral load, then you get a direct correlation. It means higher the level of galactin 9, higher will be the HIV viral load. It means that a galactin 9 is pushing or stimulating HIV infected cells to release or a lot of viruses, or viral replication is enhanced. We use this therapy for a latency reversal agent, which it's not part of this lecture today, but it has a different, what you say that, uh, application which we have been exploring. Now, after eclipse, early responses we are catching 10 days is it eclipse period where you don't see the RNA at all, and then followed by that we started observing different cytokines. Galectin 9 came 5 day post viremia. After that, there is a time for the peak viremia. If you recall that the virus was going up, 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 and after reaching at the top, then they started coming down. So when they reach to peak viremia, why it is happening? Because dendritic cells took the HIV, took it to the lymph node, and in lymph node it infected, activated CD4 cell, which is a CD4 positive, CCR positive T cells, and now it is enhanced by dendritic cells. Important thing is that since Lymph node is a target, final destination of HIV, where it starts, where it goes and it starts infecting other things. The hub for lymph node, where maximum number of lymph node are present, is your mesenteric lymph node, the lymph node which is associated with your gut. You see this picture here. You can see several blue spots, which we call mesenteric lymph node, isolated lymphoid follicle pair patches, and this all together are termed as gut associated lymphoid tissue. Lot of CD4 cells are there, and we call it gut associated lymphoid tissue. So ultimately, virus goes to gut associated lymphoid tissue. Here, the interesting story comes. Inside gut, which is gut associated lymphoid tissue, approximately 20% of CD4 cells are infected, and 60% of uninfected CD4 cells becomes activated. See that if there are 100 CD4 cells, 20 will get infected, and because they get infected and get activated, because of the activation, they also activate other 60% of CD4 cells through cytokines, chemokines, and some other molecules which people are still investigating. So, total 60 plus 20, it becomes 80. So, 80% of CD4 cells in the gut associated lymphoid tissue, it's depleted within 3 weeks of HIV-1 infection. And virus level reaches around a million copies per ml within 21 and 28 days, right there when the virus is at the peak. Now this is the time when your antibodies are there, CD8 cells starts coming out, and then your viral load starts coming down. And this is just a overview how actually it looks like, because gut associated lymphoid tissue is associated with your gut. And if you look at the gut HIV negative, there are so many villi inside it, which is a good. It helps in the blood processing and all that. But HIV positive subject, you see this so smooth. So it looks very good, smooth, but it is not good, because the smoothness is not what we want inside our elementary canal, because it has to grind the food properly. And why it has happened? Because they lost some kind of villi, and as a result of which all these epithelial cells you could see here associated with the gut, they develop a tight, they develop a gaps between two epithelial cells. When they develop gap between two epithelial cells, then the the intestine or gut becomes porous. This is called leaky gut, which I will discuss later in the slide. So this is what we call leaky gut problem, because of the disruption of the tight junction existing between two epithelial cells in HIV infection. This is possibly because there is a massive depletion of CD4 cell in the intestine. Now, once there is a depletion of CD4 cells and there is a disruption of tight junction existing between two gut epithelial cells, then gut becomes porous. When gut becomes porous, then all the bacterial population that is existing inside our intestine, it starts translocating from intestine to your peripheral blood. So when it comes to your blood, so what we should expect? All these bacterial population, if it is gram negative, it will have lipopolysaccharide. And it means that in the plasma of HIV infected people, we expect to measure a high level of lipopolysaccharides. So you see that three different categories of individual. Here you see HIV negative people. On the extreme right, we have untreated HIV infected subjects. The median, you can see that it's a bit much higher LPS level compared to those who are on antiretroviral therapy. Median significantly drops down, but it is not normalizing to that of HIV negative people, suggesting that this microbial translocation is still happening despite antiretroviral therapy. And then what happens, as I said, that you develop antibodies, you have a CD8 T cells, which controls the virus. So the viremia piece comes down and then becomes stagnant for several years. So here is the phase which we call chronic phase. It means now we have covered acute HIV infection. Now we are getting into chronic phase. Most of the HIV infection are detected at chronic phase. So you can see that there are three, what you can say, peaks. Each peak is representing different individuals. So one individual have high viremia, and then this stagnancy in this viral load. It's called virus set point. Virus set point. So you see that higher the virus set point, higher will be the disease progression. Those who end up having, for example, this third one has the lowest virus set point, it will be regarded as slow disease progression or slow progressor. So there are normal disease progressor, slow disease progressor, or higher set point means rapid disease progression. So what decides whether the person will have higher viral set point, moderate viral set point, or lowest viral set point? Because virus set point is very important in chronic infection. If you have high viral set point, you will be treated as a progressor. If you have very low viral set point, you will be treated as a low progressor. So, or you can also be called as a controller. So, there has to be some immunological parameter that controls the viral set point. So, there we need to understand what actually is happening. Now, we have HIV infected cells. HIV infected cells express or present HIV antigen on its surface. This is detected by some T cells, and these T cells we call HIV specific T cells. Now, the job of this HIV specific T cell is to expand and kill HIV infected cells right here. But it doesn't happen this way, because once you are in the chronic phase, virus keeps mutating. When these T cells kills one cells, two more cells are generated with a different epitope of HIV, as a result of which these T cells keep expanding. When they keep expanding, during this process, they are continuously exposed to or continuously stimulated by the viral antigen, as a result of which they get exhausted. You see that the person is sweating because the person is exhausted. Likewise, our T cell also get exhausted. So it leads to incomplete clearance of HIV infected cells, leading to immune exhaustion. One of the important parameters of chronic viral infection as well as cancers, where you have the disease in chronic phase, your immune system is completely helpless because immune system is so exhausted, and then it fails to eradicate the disease. This is what is happening during the chronic phase. So what happens when HIV infection is there in chronic phase? These four features are there which makes the entire things complicated: inflammation, immune activation, immune senescence, immune exhaustion. Four characteristics of the immune systems which doesn't let disease to go away. If we are able to normalize all these four things, then disease will go away. So most of the research around everything, almost everything, whatever people are doing, is focused on normalizing these parameters so that there is a better outcome of HIV infection. I hope you guys are with me. I just take a sip of water.
Yes, yes, all are here.
कोई चीज ऐसी वैसी लगे तो पूछ सकते हैं.
हाँ हाँ, कोई
वो-
क्वेश्चन हो तो आप पूछ सकते हो और इस चैट बॉक्स में लिख सकते हो.
जी.
यस सर, प्लीज कंटिन्यू कर दीजिए.
So these immune activation, immune senescence, immune exhaustion, inflammation. Now, what are these parameters? And mind you, these parameters are also valid for cancer, because cancer is also chronic. You live with cancer for several years. You live with HIV for several years. You live with HCV, hepatitis C virus for several years. You live with hepatitis B for several years. So these parameters are common in all the other infection or even cancer. So markers of T cell dysfunction and all these features which I mentioned, immunosenescence, immunoexhaustion, immune activation, they cause T cell dysfunction. Because when you reach to chronic phase, you have two arms to fight with an infection. One is your T cells and another one is B cells. I am a B T cell person. uh, I believe that T cell is more important. If you know a little bit of immunology, so you will you must have heard that the T helper cells. The name helper cells is designed because, or is coined because, it helps B cells to produce neutralizing antibody. So it means that if there is no T cell, then the B cell will not be good enough to produce neutralizing antibody. So everything is focused on the T cells here. So immunosenescence, how you will find out whether you have senescent T cells or not, it is described by the presence of a marker CD57 and CD28 negative. Immune exhaustion, there are so many molecules which we call immune checkpoints. These are TIM3, CTLA4, PD1. There are also more markers for like TIGIT. There are LAG3, right? And immune activation you can detect by looking at two important markers. One is called HLA-DR and CD38. So when you want to study the level of immune activation, you will stain your PBMC or T cells with these two antibody specific to these two markers. And if they are highly expressed, you say that the person is having high immune activation. It means that's not good. He's leading to, it will lead to the rapid disease progression. So we have to think about how to reduce it. Likewise, you have high level of immune exhaustion. All these markers are elevated. It means that your immune system is completely dysfunctional, at least adaptive immune system, and the person is going to go to this rapid disease progression. And immunosenescence is also one thing. As we age, our T cells by default get into the senescent phase. It's called immunosenescence. But in HIV infection, what happens during aging in normal healthy people, during HIV infection, it happens quite early in their life. That is why accelerated aging is a hallmark of HIV infection. So people also uh, study or do research, accelerated aging aspect in HIV and during HIV infection. That's another important topic. Now, when we talk about the good T cells, which will control the viremia and suppress the virus progression or disease progression, so we need to know these parameters. These are important molecules which are released by different kind of CD8 T cells, which makes them cytotoxic or cytolytic T cells. They are perforin, granzyme, cytokine, and fast ligand. So T cells will be effective if it has all these features, and when it has all these features, then it is very easy to control the virus. Here are some of the examples how these markers control the disease progression. I just told you in the beginning, the elite controllers are the group that controls the viremia by themselves. So it's interesting to find out what is actually happening in this. So when you do immunophenotyping and we just gate, when you gate your CD8 T cells against perforin and interferon gamma, then you will get to know that all the HIV infected cells, which is interferon gamma producing cells, this elite controller have the highest proportion of perforin. So it means that HIV infected, HIV specific cell, which is recognized on the basis of interferon gamma positive cells, they have highest percentage among other groups for perforin, as you can also see in this bar diagram. So it suggests that presence of perforin by default is much higher in HIV specific T cells and elite controller. That is why they are controlling the viruses. That's why they have undetectable viremia. Not only that, another marker which I mentioned, it's called immune exhaustion. Immune exhaustion marker, basically PD1, CTLA4, and TIGIT. You see that upper panel, it has y-axis PD1 and x-axis has CTLA4. In the lower panel, they have PD1 and x-axis has TIGIT. And different categories. It's called elite controller. ERT suppressed or HIV infected people with antiretroviral therapy. Third category is viremic, those who are not taking any antiretroviral therapy, and this is healthy control. So if you look at this population, double positive, which is CTLA4 and PD1, you will see that highest percentage of double positive cells you find in viremic. Means that those people who are not taking any antiretroviral therapy and their viral load is high, they have highest percentage of frequency of double positive, yani ki immune exhausted PD1 CTLA4 positive CD8 T cells. When these people are put on antiretroviral therapy, you see that 1.83. It means that double positive cells got reduced but didn't normalize to the level of healthy control. But interestingly, elite controller by default they have very low double positive cell, which is comparable to the healthy control. Likewise, the same thing is happening with the TIGIT and PD1. So, suggesting that the exhausted T cells by default are absent in elite controller because their level is exactly similar to that of healthy individuals. Another parameter was immune activation. Immune activation, you find the immune activation on the basis of two marker, HLA-DR or CD38. It's an interesting story I would like to mention about. It's about Professor Janice Georgia, University of California, Los Angeles. She was the first person to come up with this immune activation concept. And what she did, she started looking at, uh, you see that HLA-DR and CD38 in different categories of people, HIV negative control, then asymptomatic HIV patient, and those who had AIDS, and people who died of AIDS. Their blood samples was stored, it was collected, and then they grouped these they grouped these people who died of AIDS in two categories. One with longer survival time, another one with a shorter survival time. And it was found that shorter survival time was observed with the T lymphocyte activation. It means that shorter lifespan if there is a high immune activation. You see that more than 18 month survival, less than 6 month survival. And this is a flow plot for CD8 cell and CD4 cells. And here you see the bar diagram. Well, this is box plots. And here you can see that those with the shortest survival time, less than 6 months, that had high CD38 molecules on CD4, also on CD8 here, followed by individual who had larger survival time, which is more than 18 months, and this is the control. So immune activation is linked with the shorter lifespan among HIV infected subject. That's what she, for the first time, um, proposed, and it became so popular. There were hundreds of paper people published in chronic HIV infection, which they talked about immune activation. We did something new when I came back to India and started working on pediatric HIV, and where we looked at perinatally HIV infected children, because perinatally HIV infected has a completely different disease progression. And we had the samples from St. John's Medical Research Center at Bangalore, and we did this fantastic study where we took a two group of HIV infected children. One group was on therapy, another group was not on therapy. Those group which are not on therapy we call treatment naive HIV infected group, and we did immunophenotyping for detecting immune activation, for which the gating strategy was pretty simple. Singlet were gated against FSC height and FSC area, and then we have a lymphocyte population. I'm sorry if people are not familiar with this flow cytometry gating. It would, it would be difficult to make you understand about the concepts of flow cytometry within this lecture, because it requires a different lecture. So, but just look at these figures. Lymphocyte population was selected, and then based on live and dead marker, we ended up finding CD3 positive cells, and CD3 positive again gated for CD8 and CD4. So these two populations were further used for detecting immune activation marker, which is HLA-DR and CD38. See that the upper panel we have a CD4 T cells, and the lower panel we have CD8 T cells. In the upper panel A, you see HIV negative subjects gated against CD4 cells was gated against CD38 and HLA-DR, and you see this double positive. Here is 1.39 people, or these children who are on antiretroviral therapy, this 7.84, and those children who were never on treatment, high virus, they have double positive cells, which is 13.9. We can say that there is a high CD4 T cell activation. Likewise, in CD8 T cells, 34 double positive cells, HLA-DR and CD38, means that CD8 activation, CD8 T cell activation, it's 34.6. So HIV disease progression of viremia is linked with T cell immune activation. So we can say that, well, whatever these flow plots are there, I just represented in the form of this dot plots here. And here you can see that people who are on antiretroviral therapy, there was decline in the immune activation for sure, but it did not normalize to the level of HIV negative subject. Immune activation in HIV infected patient or children on therapy was significantly elevated compared to HIV negative subjects. So this is something which is a matter of concern, because high immune activation despite no viremia, it means that these children are on the risk of getting certain complication which we call non-AIDS malignancies. In the same children, when we started looking at different cytokine, pro-inflammatory cytokine level, so one thing which there were several, you can see that with the blue it was elevated, but one important molecule or chemokine, cytokine, it's called IP10. IP10 is a very, very important molecule, inflammatory molecule or pro-inflammatory molecule, which is associated with the disease progression. And when we found it in perinatally HIV infected children, then we also observed that this IP10 remains elevated despite the therapy. You can see that HIV negative individual IP10 level on ART median is pretty higher, p value is 0.03. So it means that IP10 doesn't get normalized
सर
despite.
और कितने स्लाइड्स हैं?
स्लाइड तो है अभी, लेकिन खत्म करता हूँ 10 मिनट
जी
में, आई
जी
विल रैप इट
जी
अप है.
जी जी जी
So
जी
IP10 is like that. So another feature is immunosenescence. Immunosenescence is defined as a decline in the function of immune system with age. And what we actually, and immune exhaustion is exactly the same thing which I just mentioned. As you can see here, what was done in chronic HIV infected subject, it was found that TIGIT and PD1, which is a exhaustion marker, is elevated in HIV infected subjects. Let's say the 53.6 compared with 7.39, and all different category has elevated TIGIT PD1, which is a double positive immune exhausted T cells. And if you try to block it with the anti-TIGIT and anti-PDL1 antibody, so you'll see that the proliferation of these cell increases. It means that blocking this exhaustion marker will revive the functionality of these T cells. So these days, therapies are being considered where you block the exhaustion molecule and then revive these T cells. We also actually looked at the same TIM3, another exhaustion molecule, and compared between children and adolescent, and we found that along with the age or duration of the infection, TIM3 positive CD8 T cells increases, and which makes the T cells what you say that uh, functionally impaired. And likewise, you see that TIM3 positive CD8 T cells remains elevated despite treatment or elite controller in HIV infected control. So I just also mentioned about we also looked at uh, exhaust senescent T cells, and we believe that people who are on antiretroviral therapy, they have normalized senescent phenotype. But when we compared our HIV infected individuals on antiretroviral therapy, we found that they have significantly elevated exhausted CD57 positive CD8 T cells, which means that immune senescence still exists even in people who are on therapy. and they people also have elevated level of IL-8 and TNF alpha, and that was correlated with the inflammatory cytokines. So it means that these inflammation, everything is there. Now the situation is why it is happening. Now we get into the core of the entire talk. What I'm saying that what is the cause of elevated inflammation despite antiretroviral therapy? So there are two major, what you say, probabilities. One is gut epithelial barrier dysfunction. Remember, I just mentioned that HIV infection disrupts CD4 cells, and after disrupting CD4 cells, they actually make a space between gut epithelial cells, and the gut becomes leaky. Now, when gut is becoming leaky, so why it is happening? There has to be some mechanism through which gut has to remain its integrity. So what is actually causing this leaky gut syndrome? We will see in the next slide. And another feature is residual HIV replication. We mainly talk about the first point. The first point is related with the HIV infection that alters the gut microbiome. So very quickly, I'll just explaining that whenever HIV infection happens, there is a change in the gut microbiome. You can see that HIV positive individual have high relative abundance of Prevotella, and this Prevotella, you can see that remains elevated despite the treatment. And when this Prevotella remains elevated despite the treatment, then what will happen? You also need to see whether there is a microbial translocation. We looked at microbial translocation on the basis of lipopolysaccharide. Here is a marker called soluble CD14, and this soluble CD14 is elevated even on treatment groups. So it means that microbial translocation is happening and Prevotella is elevated when these two things are there. And then what is the correlation? There is a direct correlation. It means higher the relative abundance of Prevotella, higher will be the microbial translocation. When microbial translocations increase, then it will interact with the your myeloid cells and other immune cells, and as a result of which they produce a lot of pro-inflammatory cytokine. So when they produce pro-inflammatory cytokines, it means that you are at risk of developing complication. So how to mitigate the high level of inflammation in your body? If simple answer is that if you are able to reverse gut microbiome to the normal level, it may be possible that you can mitigate the elevated inflammation, immune activation in HIV infected subject on treatment, and all these important feature of during chronic HIV infection, or even early phases of HIV infection. It is very helpful because it helps us to come up with a novel therapeutic intervention to control the HIV disease progression. So this is just a crisp of the massive HIV immunobiology, which I tried to explain you all, keeping everything in nutshell and simplifying it as much as I can do with my language or with my illustration. And I hope that you just got something beneficial out of this talk. Thank you so much.